will present a lot of new results today. Hopefully it's new to you. I, of course, work on these methylation-based clocks, and allow me to review slides. First of all, methylation allows you to build biomarkers with three extraordinary properties. Number one, these apply to the entire life course, really starting with prenatal development. So epigenetic clocks are life course timepieces as opposed to just aging biomarkers. The second remarkable property of cytosine methylation is that you can build clocks or measuring age in all cells that contain DNA. You can build so-called pan-tissue clocks. That's quite different for RNA or proteomics, but cytosine methylation allows you to do that. And the third property is that you can build clocks that apply to multiple species at the same time. And I will talk about it today. And as you know, a clock is fundamentally a regression model where you want to estimate either age or morbidity risk, and the covariates are cytosines. So these are three properties, and I will show you a lot of examples illustrating that. I want to remind you of some terminology which I will use throughout the talk, which is first generation clocks, second generation clocks, and third generation clocks. First generation clocks are really trained as predictors of chronologic age. So they are chronologic age estimators. Second generation clocks are meant to predict mortality risk or morbidity risk or health span. So they really predict, for example, time to death or time to another adverse outcome. And then finally, I use the terminology third generation clocks. These are, again, regression models or predictors, but they are now trained on multiple species. And now I want to briefly touch on a new first generation clock that we developed. We call it a Centenarian Clock. And you see from the title that it is meant for one purpose only, validating claims of exceptional longevity. In other words, if you know somebody who claims that they are 150 years old, and then they tell you, oh, sorry, I lost my birth certificate or whatever, we can now verify age claims. And so this was inspired by this recent controversy surrounding Jean Clement. Many of you have heard about it. And we just felt, can't we develop an epigenetic clock? And so here I show you one of two epigenetic clocks that we have developed for centenarians. This one is based on a neural network, but we also have one based on elastic net regression approach. And the exciting thing about this PowerPoint slide is the x-axis. Notice the chronologic ages here. So all of the dots correspond to blood or saliva samples from centenarians. And this is a cross-validation study of the clock. And the remarkable thing is actually the high Pearson correlation between age and the predicted value based on methylation. Correlation 0.64, which is truly remarkable given that the age range, right, goes from 100 to, I think, 115. So it's quite accurate. And in any event, there you have it. I just give you that as a flavor of this centenarian clock. And but here's an analysis that is perhaps interesting to people who are interested in the biology. So let me explain what we have done here. each dot is now a cytosine. And the x-axis shows you how this cytosine relates to age in very old people, defined as 90 plus, right? So we do what is known as an epigenome-wide association study and correlate each cytosine with age in 90 plus people. And what's on the y-axis? Well, there's the same, but now we did an EWOS in people younger than 40, okay? And what you see from the high correlation is that in general, cytosines that gain methylation in these younger people, younger than 40, the same cytosines also gain methylation in 90 plus, you know? And that brings me to this claim I started with, which is that epigenetic clocks are actually life course clocks, right? So even things that change in 90 plus also actually change early in development as well, you know. I'm making broad statement. You always find exceptions, right? You see here cytosines that have the opposite pattern. They only gain methylation in centenarians, but not in younger people and so on. But I'm just saying, in aggregate, that's what you see, preservation, pretty high preservation. I now want to come to new second generation clocks. Remember, second generation clocks predict mortality risk or morbidity risk. And I just want to announce that we have a new version of our best mortality predictor, which is known as Grimage. So we call it Grimage version 2. It actually uses the identical 1030 cytosines as the version 1 of Grimage. And but what's new? So here Akelu added methylation-based estimators of hemoglobin A1C and C-reactive proteins. So to remind you, Grimage is strictly a biomarker based on cytosine methylation. However, it's nice in that it's interpretable, that you can say, well, some of these cytosines or some of these methylation changes relate to these biomarkers that are listed here: hemoglobin A1C, C-reactive protein, plasminogen activator inhibitor 1, and smoking packets, and so on. And anyway, so Arke added these new biomarkers, refit the model, and we have now a new Grimage version 2 that has several advantages. The first is it's slightly more predictive of lifespan risk, mortality risk, and I'll show you that. The second advantage is the technical noise is reduced. And I should tell you, even the original version of Grimage really stood out in terms of very low technical noise. However, the version 2 is even more robust. You know, that's known as test-retest variability. You know, so if you draw blood 2 days in a row, then this new Grimage version is more robust. The third advantage of version 2 is it seems to apply to saliva samples. I should say Grimage was designed as a blood-based test, but version 2 seems to be applicable to saliva. And here I now show you a comparison of the new version of Grimage versus the old version. So the new version is in the left column and the old one in the right column. And personally, I only look at the red p-values. Here you see mortality prediction based on a Cox regression model in many epidemiological cohort studies: Framingham Heart Study, Women's Health Initiative, Jackson Heart Study, BLSA, Lothian Birth Cohort, Normative Aging, very large studies. And you see the p-values, when we look at everybody on the top, I mean, very significant, actually for both versions. But then the question is always, well, what about if you apply Grimage to smokers, people who smoke? Do you still predict mortality risk? And that's answered in the middle panel, yes. And then in the bottom, I show you the results for never smokers, people who never smoked. Can you use Grimage to predict mortality risk? So again, the answer is yes. Now, if you care to know, what should you do to stay young according to Grimage? And I wanted to show you the results. So the first two columns in this table show you how various biomarkers relate to Grimage 2 in the first column, or Grimage version 1. And so what do we see? I look at these, I hope you see my mouse, the mean carotenoid levels. These are blood-based markers of vegetable consumptions. And I continue to be amazed how strong the relationship is. Correlation minus 0.29, with of vegetable intake. To me, that's a strong correlation. So sure enough, I eat plenty of vegetables. Conversely, at the bottom, you see in red smoking status. Well, by design, these biomarkers pick up whether you smoke. So it's bad for you. The other thing I notice is FEV in blue. So yes, this is obviously a measure of lung functioning. It's well known. This FEV is very associated with mortality risk, and sure enough, Grimage picks that up. And you see relationships with HDL cholesterol and so on. I'll stop with that. We can discuss it later if you are interested. I now want to move to a different topic, which is third generation clocks. So remember, these are regression models that are estimating chronologic age in multiple species at the same time. And I'll start with our primate clock. So primates evolved 55 million years ago. And so in theory, these are vast evolutionary distances. And it seems to be difficult to build a clock or one regression model that measures age in all these primates. But I can tell you, it was actually really easy. It was easy because we used a special array, which I call the mammalian methylation array. If you're interested in it, it's available to all of you through my nonprofit Epigenetic Clock Foundation. The array looks at highly conserved cytosines. And so, yes, we applied these methylation arrays to 37 primate species. Again, these are cross-validation estimates of the clocks. The middle panel shows you the performance in humans. And you see correlation 0.98. So yes, the primate clock is very accurate in humans. In the lower right, you see the application to lemurs. Again, these are separated by us by 55 million years of evolution, and you still see a correlation of 0.9. And so the clock applies to baboons, rhesus macaque, marmosets, really all primates that people care about. Now I want to come to what we call universal pan-mammalian clocks. This work is again spearheaded by Arkelu. And that was facilitated by our mammalian methylation consortium. Almost 200 investigators contributed in many different ways. And here I show you a clock that is based on almost 12,000 tissue samples from 300 plus species. And a single regression model is now used to measure aging in all of these species. The right panel shows you the mathematical trick that we use. Carefully look at the x-axis. it's a number between 0 and 1, an age measure of between 0 and 1. That measure is called relative age, which is defined as the ratio of the age in years divided by the maximum lifespan of the species. So that ratio takes on a number between 0 and 1. And then the y-axis shows you the methylation-based estimate of this relative age. Again, it's a cross-validation study, and you see a correlation 0.95. And to me, this is really, again, this miracle of cytosine methylation, you know, that you can do that. Now, here I show you how this universal pan-mammalian clock applies to different species. The upper left panel shows you humans. Correlation is remarkable, 0.96. Then upper middle panel, we apply this clock to mice, then to dolphins, dogs, elephant, bats. So yes, the clock is quite accurate. By the way, it's not as accurate as a species-specific clock, you know. So if you really care about measuring aging in dogs, we have a dog-specific clock for you. But what is interesting that even the pan-mammalian clock leads to a correlation 0.94 in dogs, you know. All right, now talking a bit about this aspect that whatever you observe late in life is preserved in young animals. And so just want to briefly touch on that. So again, here we do an EWOS study. Each dot is a cytosine. And it's now correlated to age in all of these different species. And the x-axis, for example, if we focus on the rightmost panel, the x-axis shows you how each cytosine relates to age in old animals, where old was defined as a multiple of the age of sexual maturity. Old was defined as 3.5 times age at sexual maturity or puberty. So in any event, and the y-axis shows you the association of each cytosine with age in young animals, defined as less than 1.5 times age of sexual maturity. And what do you see? Strong conservation, you know. So that's, again, when you deal with cytosine methylation, you can find subsets of cytosines that relate to age consistently in development and old age. Now I want to, so this, again, this is a pan-mammalian clock, right? Which is, again, to me, very surprising that you can even do that. But now you could try to become greedy and you can say, well, can I do something more ambitious? Can I build a clock that works in a mammal and also in a frog, in an amphibian animal? And so here I show you results from Joseph Zoller and Christoph Niers, who looked at Xenopus, which is the so-called African clawed frog, which interestingly is widely used in the biomedical research community. And so let me start out by saying we had multiple tissues from clawed frogs from Christoph Niers' lab. And here I show you, yes, we can build a clock for these. Now, you may not be impressed by the correlation of 0.88, but I want to tell you why it's exciting. The reason is because we applied the mammalian methylation array to that species. So the mammalian methylation array was designed for mammals, but it contains several thousand cytosines that also are applicable to this clawed frog. And so yes, it worked. You know, you could still generate meaningful data. But here I show you now the even more challenging analysis. So Joseph Zoller now developed a clock that applies both to humans and to frogs. And the left panel shows you in black dots are human samples, just regular human samples, and the colored dots are the frog samples. And now the right panel, so this is what we call a dual species clock. It applies to humans and it applies to these clawed frogs. And on the right panel is an excerpt, really. And I mean, the correlation is still quite impressive, 0.86, so yes, somehow one seems to be able to cross that species barrier. If my memory serves me right, we diverged from clawed frogs about 300 million years ago. I mean, it's a vast distance. Now you can ask, well, which cytosines relate to aging in the clawed frogs? And we find similar things that we find also in mammals, which is genes that play a role in development. And here I show you this comparison where on the x-axis we relate cytosines to age in the frogs, and on the y-axis we relate cytosines to age in mammals across all mammalian species. And to me, it's quite impressive that, yes, you can find cytosines, that are consistent between these very different species. There are obviously there are plenty of outliers, only 0.26, so don't walk away from this talk thinking that everything is beautifully conserved. It's not. It's rather the other way around. You can find subsets of cytosines that show conservation, but most cytosines do not show conservation. All right. Now, as you know, I certainly like epigenetic clocks, but we need to acknowledge what I call original sins of the construction of epigenetic clocks, drawbacks. And the first is there's really a multiplicity of clocks, many, many clocks. Especially we have now dozens of mouse clocks, for example, and many human clocks and so on. And the multiplicity arises because you can analyze and develop clocks on different training sets, different filtering approaches, many ways of building a clock. And the second, and that of course challenges us with communicating results, because in a worst case scenario, a finding can be only obtained with one clock, but not another clock. And so we need to find strategies of applying multiple clocks to the same data set to convince us that the results are robust. And the second original sin is that biological interpretations of these clocks really requires additional work. Why? Clocks were defined on the basis of a mathematical optimization procedure, right? For example, least squares regression or any other way of optimizing a machine learning algorithm. So you can build a black box predictor, but you need to then do the work to learn the biology. But fortunately, many labs have worked on the biologic interpretation, and I think we know quite a lot about that. So let me start out by, again, this topic of multiplicity of clocks. We have many clocks. How do we decide which clock is good, which one is bad? Well, by developing gold standard benchmark data. And we have that now. So in mice, we have benchmark interventions where I would say if your clock doesn't show that the expected result, then you kind of need to explain why not. So for example, we have well-known pro-aging interventions, high-fat diet in liver, in a mouse, for example, or even in humans. Obese humans show strong epigenetic age acceleration in human livers. So both humans and mice. Similarly, trisomy 21, which is known as Down syndrome. Again, both humans and mice show that. So that's pro-aging. Now, anti-aging, caloric restriction in mouse livers, and also these dwarf mice, growth hormone receptor knockout, multiple tissues. And I wish I had added it on this slide. We recently analyzed Snell mice. That's a collaboration with Rich Miller. And but this is known, by the way, many people who are aficionados of mouse clocks, they know that these dwarf mice show also delayed epigenetic aging. And the third intervention I now added is parabiosis. So yeah, in my worldview, if you have a mouse clock, it would be nice if it showed the expected relationship based on these benchmark interventions. So now I want to come to this point of biologic interpretation. And I briefly want to draw your attention to an article by my collaborator, Ken Raj, who published this article on hallmarks of aging related to epigenetic age. And Ken really focused on comprehensive in vitro studies. And I don't have the time to go into the details, but I want to show you the punchline of his extensive studies. He said epigenetic clocks relate to deregulated nutrient sensing, mitochondrial dysfunction, stem cell exhaustion. Today, I want to focus on this last bullet point, altered cell-cell communication. I have here a picture of a vampire because I wanted to be funny, but basically this idea of young blood, somehow you inject young plasma, or these parabiosis experiments that I just mentioned, where an old mouse is connected to a young mouse, or this idea of using extracellular vesicles. And I will talk about 4 studies that used epigenetic clocks to some detail, because I feel this kind of insight deserves attention. That yes, this young blood and whatever is in the young blood, which conveys this cell-cell communication, that yes, that affects epigenetic clocks in the expected ways. And I start with a study from Harold Ketcher and Akshay Sanghavi and many others, where Harold developed a young plasma fraction. And he injected that in rats. So these are, I want to maybe emphasize the sex of these animals. These are now male Sprague-Dawley rats. Why do I emphasize the sex? Because many treatments have sex-specific effects, right? We all know many anti-aging interventions work only in male mice, but not in female mice, or the other way around, So so Harold looked at these male Sprague-Dawley rats, injected plasma fraction into old rats, and we analyzed multiple organs of these animals using now a 3rd-generation clock. This is now a human rat clock. And what you want to compare are the red bars. These are the epigenetic ages of the old rats. And you want to compare to the orange bars. These are the rats that received this young plasma fraction. And what do you see in blood? Left panel is blood, drastic rejuvenation. Liver, drastic rejuvenation. Heart, drastic rejuvenation. Hypothalamus, less so, but maybe also rejuvenation, So to me, these were stunning results. But that's just one study, male rats. Now I want to draw your attention to another study, Parabiosis study by James White, Vadim Gladyshev, Bohan Zhang, and many others. And they look now at female black 6 mice, different gender, females, different species, mice. And they parabiosed these animals for 3 months. These animals were connected. And what was particularly interesting in this study is that then they detached the mice for 2 months. Why is that important? Because on certain levels, after 2 months, the blood is washed out, right? They went on leading their separate lives. James, they harvested these organs, and they applied many epigenetic clocks, and they all showed the same thing. Here I focus on the clock experiments that we did with the universal mammalian clock, the 3rd generation clocks, in the upper panel. And what do you see? Even after basically rejuvenation in the expected way, if an old mouse was connected to a young mouse, then sure enough, the organ was rejuvenated. And we saw that after 1 month. We saw it after this detachment. As I mentioned, these animals were detached for, let's say, 3 months. And again, the rejuvenation persisted, And I briefly mentioned the opposite could also be observed. If you had a young mouse, and you attached it to an old mouse, then the organs of the young mouse showed accelerated epigenetic aging. So based on that study, I really elevated parabiosis as a gold standard intervention for epigenetic clock evaluations. So that was that. I want to now come to yet another study by a totally different team, Jorge Sanz Ross, Consuelo Boras. This is a Spanish team. And they evaluated small extracellular vesicles from young mice, and they injected it in old mice. Again, to me, this is under this heading, young blood. And so maybe to remind everybody or to introduce you, so extracellular vesicles are released by virtually all cell types. But you can then focus on extracellular vesicles from stem cells. And so the authors here looked at stem cell derived extracellular vesicles, and these came from adipose derived stem cells. And they injected them in 2-year-old mice. Sorry. Here I just want to outline the experiment. So there was a baseline measurement, and then they injected 2 times these extracellular vesicles, day 1, day 7, 2 injections. And then they harvested the organs at day 30. So 1 month later, the animal was sacrificed. And we looked then at multiple tissues with our epigenetic clocks. We have many mouse clocks, as I won't mention, I won't go into them. But what I can tell you is, as you can see, we saw rejuvenation in kidney tissue and also in liver tissue of these animals, but not really in muscle or spleen, So in any event, I like these results because they added to this story, right? Parabiosis, a rat study. And so I just thought it's interesting that we saw that here as well. I cannot quite give justice to this study because the collaborators looked really at many other readouts, grip strength, motor coordination, many other biomarkers, But anyways, I focus on the clocks. So we have strong evidence to summarize this study that in rats and in mice, young blood rejuvenates various organs. But what about humans? And here I want to draw your attention to a study from James Clement, who many of you may know. He conducted a relatively large phase 1 clinical trials where he looked at umbilical cord plasma concentrate. And I'll show you this study. So James evaluated 18 people. This was a phase 1 study. It's meant to establish safety. But the punchline is we also saw that it reversed Grimage. But so just to tell you, so these 18 individuals received these plasma injections once per week for 10 weeks. And then James evaluated almost, I think, 78 clinical biomarkers to establish safety. But then we also applied our epigenetic clocks. Now, these people, 10 women, 8 men, and ages range from 60 to 95. And here I show you in the right panel, in certain ways, our best result, which is that this Grimage clock, which is our best mortality risk predictor, showed evidence of rejuvenation after that treatment, and p-value is 0.001. And now Grimage, as I mentioned, is based on a combination of methylation-based estimators of various plasma proteins. And so the left panels here show you methylation estimators of growth differentiation factor 15 and cystatin C, and you again see a beneficial effect. And so yeah, because I just want to now discuss these results from the young plasma study. So many of you know there's a large body of literature that shows that young blood plasma or young blood has beneficial effects. It goes back decades. Many names should be mentioned. Many people. Now, James Clement did the first human study that investigated umbilical cord plasma concentrate and its relationship to epigenetic age. And I mentioned this plasma concentrate, these intramuscular injections of 1 ml were well tolerated. We found nominally significant age reversal for the Grimage clock, but I need to add the other clocks we have did not show a rejuvenation effect. So pan tissue clock, for example. So first generation clocks did not show age reversal. You really needed Grimage and a second generation clock. Then, as I mentioned, we found also evidence that various clinical markers were rejuvenated, especially clinical markers related to kidney function, for example, cystatin C levels. Yeah, so that's the end of that story about cellular communication and epigenetic clocks. I think the evidence is quite strong that there's an effect. Today, I didn't talk about another topic that I present quite a lot, which is this study of mammalian lifespan. Our mammalian methylation consortium, 48 mammalian species, and we managed to build estimators of maximum lifespan. Here I show you a predictor from Cesar Lee in my lab. But also we characterize cytosines that relate to mammalian lifespan. What I can tell you is there's a very strong relationship, which again shows you that a cytosine methylation is quite interesting for longevity, maximum lifespan, and so on. And I need to acknowledge many collaborators. I think I mentioned several names: Akelu, Armin Hagani, Cesar Lee, Joseph Zoller, many collaborators. I mentioned that we distribute our mammalian array through a non-profit headed by Bobby Brook. And I'll stop with that. Thanks so much.